It is my great pleasure to introduce Dr. Gabe Zentner. Gabe is a PI, an assistant professor at Indiana University in Bloomington, and he did his postdoc with Steve Henikoff at Fred Hutch before I got there. And he did a lot of great work uncovering mechanisms of transcription in collaboration with Steve Hahn's group, and he's really developed the STRIPE-seq method, which I think has been used a lot more recently since Gabe's work in the Henikoff and Hahn labs. And I'm really excited to hear what he has to say for what he's been doing in his own lab. And with that, I will leave the video for Gabe.
All right. Everybody hear me okay? All right, so thank you for the kind introduction, Christine, and thanks for having me on to speak. And thanks to Apurva for giving a great talk to start. So we'll keep on the transcriptional theme, but we will stick to initiation. We're going to hang out at the promoter for my talk. And I'm going to be talking about a new method that my lab has developed for precise mapping of transcription start sites that we call STRIPE-seq. I'll tell you what the acronym means later, but for now it's simply given me another bad pun to put in my talk title. So I like to start a discussion of transcription start site mapping with the textbook view of the eukaryotic promoter. I'm not going to go through this in any great detail. This is just your standard yeast promoter. We can debate about whether there should be a Tata box there or not. Same with a metazoan or multicellular eukaryotic promoter. We have our various core promoter elements. We have our enhancers and insulators rather far away. But what I want to focus on for the purposes of my presentation is the black arrow here at the +1 base position. This would be the transcription start site for this yeast transcription unit. This black arrow would be the transcription start site for this metazoan unit. However, I would argue that this textbook model of transcription initiation, that it starts, transcription starts at a single position for every gene, is inaccurate in most cases. In fact, heterogeneity of TSS or transcription start sites within promoters can have wide-ranging biological consequences. Transcription factors and other proteins can specify the usage of alternative promoters for given genes, and this can lead to shortening or extension of the 5' untranslated region, which can have various consequences that I'll talk about. One of the most obvious consequences for alternative transcription start site selection is that you can generate transcript isoforms. Your favorite gene 1 is the long isoform here. Your favorite gene 2 is the short isoform. And this transcript isoform selection can lead to proteins with or without critical domains. And this has been really well studied in plants. Now, beyond the production of variations in the coding potential of a given transcript, alternative start site selection can have profound consequences for translational efficiency. You can have the inclusion or exclusion of internal ribosome entry sites, thus bypassing cap-dependent translational initiation. You can have the inclusion or exclusion of upstream open reading frames or UORFs, which encode very short, usually less than 10 amino acid polypeptides that essentially act as sponges to prevent ribosomes from reaching the primary open reading frame for a given transcript. You can have inclusion or exclusion of secondary structures, and in some cases, you can just exclude all of these regulatory features to make a very short 5' UTR. Of course, 5' UTRs have a big role in post-transcriptional regulation of gene expression as well. These secondary structures that may be included or excluded can create binding sites for RNA binding proteins, and without those binding sites, the message could be destabilized due to sequence elements or what have you. So the selection of transcription start sites is really a linchpin in terms of transcriptional initiation and can have wide-ranging consequences for subsequent steps of gene expression. I'll give you a couple examples of where we see this throughout biology. A really striking and large-scale example of alternative initiation happens during zebrafish development. So what we're looking at here is a time course of CAGE signal. CAGE is a technique for mapping transcription start sites I'll tell you a little bit about later. So we're looking at genome browser tracks for a rather short segment of the zebrafish genome encompassing the upstream region of the CCNI gene. So from the unfertilized egg to about 6 hours of post-fertilization development, the transcription start site or start sites for the CCNI gene are in this maternal dominant position, indicating that the maternally deposited transcripts from you're in the oocyte in the fertilized egg initiated predominantly at this downstream position. However, when zygotic genome activation occurs at the maternal to zygotic transcription, we have this shift where the TSSs for this gene go 40 to 50 bases upstream. And in addition to their large spatial shift from the maternal TSS position, we actually see a spreading out, broadening of this cluster of transcription start sites, which we refer to as transcription start regions. And this is just one example, but the study from the Carnici lab that identified this shifting behavior described this behavior at over 900 distinct transcripts. So it's a widespread phenomenon, even though the functional consequences aren't super well understood at this point. So I'll just mention us humans real quick. It's been shown via a large-scale computational analysis that alternative initiation, along with alternative termination, is actually the primary driver of transcript isoform differences in the vast majority of human tissues. So we often think of alternative splicing as being the dominant contributor to transcript isoforms, but initiation and termination are not to be underestimated. And that's normal physiology, but in cancer and also other disease states I should mention, there is actually a pervasive rewiring of transcription initiation that, again, could have various consequences on later stages of gene expression. So I hope I've convinced you that heterogeneity in TSSs leading to these transcription start regions, or TSRs, is a very important biological phenomenon that influences most stages of gene expression. So naturally, we'd want to map on a genome-wide scale where all these transcription start sites and the transcription start regions they make up are located. And indeed, there are a lot of techniques that have been developed to do just that. So I'm going to introduce you to the alphabet soup of TSS mapping technologies that currently exist. So perhaps the most well-known method that I mentioned before is CAGE, or cap analysis of gene expression. The most recent is this no tagging, no PCR sequencing-based CAGE called nanti-CAGE. There are other iterations for low input called nano-CAGE and slick-CAGE. And then we have Rampage, another tortured acronym before I introduce you to our own tortured acronym. So these methods vary in terms of their input requirement, how expensive they are, how long they take to perform, and the quality of information they give you. So nanti-CAGE is considered the gold standard, but you need to have quite a lot of total RNA input. It's also quite expensive per sample. It takes a full work week, but it gives you exceptionally high resolution. Similarly, Rampage takes a lot of total RNA. It's cheaper per sample, it's a bit shorter to perform, and it gives you somewhat lower resolution. So as we get into these lower input methods, a nano cage with two different amplification approaches, the cost is quite reduced. The RNA input is also quite reduced. They're a lot quicker too, but they have relatively low spatial resolution. They tend not to capture the breadth of TSS heterogeneity that we know is there. And most recently, slit cage allows you to get high resolution data with 1 nanogram of total RNA, but it's a very complicated and long protocol and is quite expensive. So as Christine mentioned, I am descended from the Hennekoff tree of science. So I'm very interested in methods development. And in terms of investigating biological questions surrounding TSS selection, I was sure we could come up with an easier, faster, and perhaps cheaper way to generate high quality maps of TSSs across the genome. And this led us to develop what we call Survey of Transcription Initiation at Promoter Elements by High-Throughput Sequencing. There's our tortured acronym. We call it STRIPE-seq. So the bullet points here are that STRIPE-seq is a very rapid protocol. You can do it in about half a standard working day. It's quite simple. There are 3 enzymatic steps. We first digest uncapped RNA with terminator exonuclease, or TEX. We do template switching reverse transcription, and then a library PCR. And in between, we perform bead cleanups to clean up the reactions and perform size selection. It's a pretty cheap protocol. My student Bob Policastro, who developed the method, calculated it at less than $12 per sample. And just the basic protocol we have developed can be used with as little as 50 nanograms of total RNA. So I just want to briefly mention how template switching reverse transcription works, because it is the enzymatic mechanism that is sort of the linchpin of this technique. So we randomly prime mRNAs and other RNAs from total RNA with a reverse transcription oligo containing an Illumina P7 adapter. So this is primed via a random pentamer. We perform full-length first-strand cDNA synthesis. Now, when MMLV-derived reverse transcriptases reach the 5' end of their template, and especially the capped 5' end of the template, they have a terminal transferase activity leading to the addition of non-templated cytosines being added to the first-strand cDNA. This allows the annealing of template switching oligo with a ribo-G, 3 ribo-G overhang, to anneal to the first-strand cDNA. This adapter, in addition to containing the ribo-Gs, contains most of the nuts and bolts of the Illumina P5 adapter. So reverse transcriptase, once this adapter has annealed, switches templates. It now uses this oligo to finish synthesis of the first-strand cDNA, thus incorporating the 5' most base position of this particular transcript, as well as most of the necessary sequencing adapter. So we first tested STRIPE-seq in our favorite organism, Saccharomyces cerevisiae. I'll show you a few pieces of evidence that we are indeed detecting legitimate transcription start sites. So shown here is a genomic distribution bar plot of one replicate of yeast STRIPE-seq. We've broken down the transcription start sites by their normalized signal, their strength. We find that the vast majority of TSSs, regardless of their strength, can be found in annotated promoter regions, though the promoter fraction is higher for stronger TSSs, with weaker TSSs slightly less likely to be associated with promoters. And if we plot the density of TSSs detected by yeast STRIPE-seq relative to annotated gene starts, that being start codon in the case of a protein coding gene, or the first transcribed base in the case of non-coding RNAs, we say that we get a very strong enrichment of TSS positions just upstream of the annotated gene start. Again, suggesting that there's quite a bit of heterogeneity. It's not just a single position where all the TSSs are falling. To give you a visual example, I'll show you some tracks. So we performed yeast STRIPE-seq with 50, 100, and 250 nanograms of total RNA. I'm not showing it here, but the different amounts of input as well as replicates of the same amount give very highly concordant results. We also did matched mRNA-seq, poly-A-RNA-seq for comparison. So we're looking here at GCN5, transcription factor, whose translation is induced in response to amino acid starvation. And what we see is that STRIPE-seq signal with each position representing a position of 5' end of a read, about 500 to 600 bases upstream of the annotated start codon of GCN5. And consistent with transcription actually initiating this far upstream of GCN4, we do see RNA-seq signal from just downstream of this TSR, this transcription start region, all the way through the end of GCN4. This is consistent with old work from Alan Hinnebusch's lab, demonstrating that the 5' UTR of GCN4 has 4 of those upstream open reading frames that essentially act as ribosome sponges to prevent this message from being translated under non-starvation conditions. So I mentioned there are many iterations of cap analysis of gene expression, or CAGE, that are treated as the gold standards for TSS mapping. So naturally we wanted to compare STRIPE-seq to these various CAGE approaches. So this is a very simplistic summary of the more rigorous analysis we did, but what I want you to focus on is that the STRIPE-seq signal, we have a couple of very strong positions here, and then dispersed weaker positions flanking them and between them. We see that for 50, 100, and 250 nanogram STRIPE-seq, and this pattern is quite similar to what is seen with slick CAGE, that super low input variation of CAGE. We see most of the same positions represented. However, in the nano-CAGE approaches, we see very few or even only one TSS position, depending on how much input was used. And this is emblematic of what we refer to as a loss of complexity in terms of TSR detection. We're only getting a few TSSs in this transcription start region with nano-CAGE, while we're getting many with slick CAGE and STRIPE-seq, thus giving us a more accurate picture of the distribution of transcription initiation. So we also wanted to see if, in addition to just giving us steady state maps of transcription initiation, STRIPE-seq could also tell us something about differential regulation of transcription initiation. So we hit the yeast with diamide, which is a thioreactive oxidizing agent. We detected about 2,000 differentially regulated TSRs. Approximately half were increased and half were decreased. Just showing you a few examples here. So we have a region of the genome which has HSP150. It's a protein that associates with the cell wall and is upregulated in response to stress. We see that STRIPE-seq signal is moderate in control conditions and is markedly increased when the cells are stressed. And this corresponds to increased total RNA or poly-A-RNA-seq signal. And at the divergent cis3 gene, we see the opposite phenomenon. Initiation decreases, as does overall expression of this gene. And second, we have a divergent pair of ribosomal protein genes, both of which display reduced initiation and reduced transcript levels upon diamide treatment, which is consistent with previous studies showing that protein synthesis is largely repressed in response to oxidative stress. To drive that home, we did gene ontology analysis of the genes associated with transcription start regions that were differentially used between control and diamide stressed cells. In the TSRs that showed a decrease, we see terms related to ribosome biogenesis and translation, consistent with the global suppression of protein synthesis during this type of stress. And for the increased TSRs, we see things we might expect like oxidation reduction process and various other metabolic processes that will help the cells respond to this oxidative stress. One more thing I'd like to mention about STRIPE-seq is that in addition to providing information on initiation, we thought because we're using paired-end sequencing and thus have fragments of transcripts, we thought maybe we could use them to actually quantify transcript levels instead of simply only using them to quantify initiation. So in order to do this, we treated paired-end STRIPE-seq data like RNA-seq data. Instead of considering the 5' most position of a sequenced fragment, we considered the whole fragment. So when we quantified transcript levels via STRIPE-seq and RNA-seq, with and without diamide treatment, we got about 1,200 upregulated genes and 1,300 downregulated genes. The majority of them were shared between STRIPE-seq and RNA-seq, the center of the Venn diagrams here. However, RNA-seq detects quite a bit larger number of differentially expressed genes. We think this is probably for a couple of reasons. We're using orders of magnitude more input for RNA-seq, and RNA-seq can give you multiple fragments, sequenced fragments corresponding to a transcript, whereas STRIPE-seq only gives you a single fragment per transcript. And we also found that STRIPE-seq was less sensitive toward genes that had a lower fold change. Despite those potential limitations, we found that the gene ontology for upregulated and downregulated transcripts with RNA-seq here on the left and STRIPE-seq here on the right were very similar. Ribosome biogenesis is down, metabolism is up. So we think you can get more bang for your buck with STRIPE-seq. You're getting initiation and transcript abundance. So that's all well and good in budding yeast, which has a relatively small transcriptome. So of course we wanted to see if our method would work in a much more complex transcriptome, and so looked at STRIPE-seq in K562 erythroleukemia cell line. It's a very commonly used cell line, and folks have done a comprehensive comparison of TSS mapping methods in these cells. So I'll take you through a few of the quality controls we did. Just like in yeast, the vast majority of TSSs, regardless of strength, can be found in annotated promoter regions. And relative to annotated TSSs, the TSSs we detect by STRIPE-seq very sharply peak right on. And if we look at a single example, looking at the TBPL1 gene, we see that STRIPE-seq signal piles up just upstream of this somewhat shorter intron, and the RNA-seq confirms that we are detecting a slightly shortened form of this particular exon for this gene. So we didn't do any treatment of K562s, so for a measurement of transcript abundance, we simply correlated RNA-seq and STRIPE-seq transcript abundances, and we get quite good Spearman correlation coefficients between RNA-seq and STRIPE-seq. I'm getting about a 0.82 to 0.84 correlation. So the measurements, while not exactly the same, are highly similar. And we lastly compared K562 STRIPE-seq to Nanticage and Rampage, two of those more established methods, as well as NanoCage XL, just a further iteration of that low input method. And to cut this correlation heat map short, what we see is that the correlation of STRIPE-seq with Cage, shown here, 0.79 to 0.8, and Rampage, 0.77 to 0.8, is as good as the correlation between Cage and Rampage, which is about 0.77 to 0.8. So we have very high correspondence between STRIPE-seq and the currently accepted gold standard methods. So the last thing I want to tell you about STRIPE-seq is that we think, even using total RNA, that we are detecting potential enhancer RNAs. So knowing that enhancers can be transcribed, some at least, we were wondering if we were detecting any transcription start regions that are far enough away from an annotated TSS that they could potentially represent enhancers. So through a systematic analysis, we identified 513 of these distal TSRs that were present in all 3 of our K562 STRIPE-seq replicates. And if we plot a STRIPE-seq signal around the center of these distal TSRs, we can see signal in STRIPE-seq, we see it in CAGE, we see it in RAMPAGE, and we see it, though less precisely, in NanoCAGE XL. So this doesn't seem to simply be an artifact of our method. And knowing further that enhancers have a characteristic histone modification signature, they tend to be enriched for histone H3 lysine 4 monomethylation, while being depleted of trimethylation at the same position, and enriched for H3 K27 acetylation, we obtained cut and tag data from a recent publication from the Hennekoff lab and investigated its enrichment at our distal TSRs. And we see that a respectable fraction, maybe a quarter to a third of these regions, display the canonical active enhancer histone modification pattern. And just to close, I'll show you a couple examples of that. We have tracks where we have K562 STRIPE-seq, CAGE, RAMPAGE, NanoCAGE, as well as histone modifications. Here, we also have, in these black bars, annotated enhancers from the enhancer DB. These were generated through integration of ChIP-seq and other genomic data sets. So we have signal in all methods, robust enrichment of both these histone modifications within this intronic potential enhancer. And here is one that is intergenic, robust signal in STRIPE-seq, CAGE, and RAMPAGE, robust histone modification signal. So we think STRIPE-seq can detect enhancer RNAs, though we are currently working on integrating nascent RNA isolation into the protocol to enhance its sensitivity. And so with that, I would like to thank, of course, everyone for tuning in. From my lab, I would especially like to thank Bob Policastro right here. Bob is the STRIPE guy. He developed the protocol. He did all the troubleshooting. He wrote all the code used to generate the figures and is currently working on a software package for TSS mapping data analysis. He should be on the postdoc market in about a year. So please let me know if you'd be interested, because he is a fantastic student. To thank our collaborators from IU and Arizona State, thank IU and NIGMS for funding, and I will be happy to take any questions.
That was excellent. Thanks so much, Gabe. You have some questions already, so I'm just going to go ahead and get to them.
Do it.
In the meantime, while I'm asking the typed out questions, if you would like to raise your hand to ask a question out loud, feel free. And if you have typed out a question but would rather ask it by voice, go ahead and raise your hand and I'll just skip over the text of your question. So to start, we have a question from Konstantin Milonis, and they're asking, does STRIPE-seq in K562 cells capture antisense TSSs at non-enhancer regions?
That's actually a good question. We didn't look at it systematically, but my suspicion is that they're too unstable for us to reliably detect in total RNA. That's something we might be able to get at by using nascent RNA, potentially in conjunction with an exosome siRNA or something like that, but we haven't really looked into it. It's a good question.
Yeah, it would be cool. I think Mike Carey had a similar question. I just see about if you can detect upstream antisense RNAs. So it'll be cool if we can look at that sometime.
We're working on integrating basically 4TU labeling into the run-up to STRIPE. So
that'll be really cool. I might want to talk to you about that at some point.
I should mention that this has been accepted in genome research. It should be online any day now. So
yeah, we'll definitely advertise it.
More to it.
Dom Helmlinger has asked, do you think STRIPE-seq would correlate even better with 4SU, or rather might detect non-productive initiation events?
Might correlate better. So since we're using STRIPE from total RNA, actually, that's not a bad idea since nascent tends to pile up. So I was not thinking about that correctly. That's absolutely possible since STRIPE-seq signal is biased towards the 5' end of transcripts, and nascent RNA signal and Pol2 chip, whether we like it or not, piles up there as well. But no, that's
in combination with other things.
we should be able to, once we've done some yeast, actually, we should just do this now. We have yeast RNA labeling data as well as STRIPE-seq, so I'm going to go bother my students after this.
That's great. Peng Lu has asked, well, first they say, nice talk, Gabe, thanks. I agree. They're wondering whether transcript length bias exists for STRIPE-seq, that is, transcription start sites of short genes, or could they be more highly represented?
That is a good question, and we have not really addressed any length bias. And the hope is that by doing random priming, we should smooth that out, but since reverse transcription only goes for so long, there probably is some fall off in terms of representation, but we haven't really rigorously addressed that.
Yeah. Toshi has asked a question. He says, great talk, Gabe. Does STRIPE-seq allow spike-in normalization?
You could do it, yeah. You could do it either with a synthesized capped spike-in, or you could do a cell mixing approach. Depends on if
that would be good.
Change it.
Especially for like stress response, it'd be really cool to be able to quantify the differences in initiation sites.
In yeast, it would be easy enough to mix Cerevisiae and Pombe like one would do for labeling.
Yeah, I'm very curious about that for my work. Harriet Kinyamu has asked, she wasn't clear about whether STRIPE can detect enhancer RNAs based on distance.
I'm not sure. There's a couple of possible distances. I mean, are we thinking based on the distance of the enhancer to the promoter, or?
Harry, if you would like to raise your hand, I can unmute you. So maybe you can ask it by voice. Okay, so they're going to do that. So I thought I saw a few more raised hands. If you raised your hand and put it back down, I will go ahead and unmute you. Okay, here we go. Oh no, wait, where did you go, Harriet? I'm seeing the hands raise and then they go to the top. Okay, there we go. Okay.
Yeah, my question was based on the distance, like the examples you showed were like pretty close to known TSSs. So I'm thinking like if an enhancer is quite far away from a known TSS.
Okay, so the criteria we established were that they could be relatively close to a TSS. 1 kb is hardly anything, but we didn't do any filtering based on distance beyond that. And in fact, this example enhancer is in the middle of nowhere. It's at least 100 kb from any annotated TSS, but we haven't looked if there's a length distribution bias at all, because we're detecting so few of them, and we wanted to be a bit more circumspect about it until we have stronger evidence, like from nascent RNA labeling or something like that.
So the reason I'm asking is because I was wondering if you could compare it to something like START-seq, if you're familiar with START-seq.
Yeah. Absolutely could.
Cool, thanks for that question. We have one more question that I didn't see at first from Natalia Kachanova, and she also says great talk. And there are some genes that you see up and down regulated in your method, but not RNA-seq upon treatment. Does that mean by deducing this overlap, you can quantify RNAs rapidly degraded from the 3' end?
That is a very interesting question, and I had not thought about RNA decay as connected to that, though that's a very cool idea that we should look at.
Yeah, definitely.
And this is total RNAs. Yeah, that's a great idea. Thank you.
Yeah, lots of really good discussions. And Sheila Tevis has one question. Does STRIPE-seq distinguish narrow versus broad TSSs?
Yeah, I didn't show it here, but we did a shape comparison, and we do see differences in the distribution. Well, I don't know if I have any really great examples here, but we do see kind of a continuum of TSR shape, if you like, from a single point source to 40 or 50 bases comprising a TSR. So we definitely see the whole range of heterogeneity.
Cool, that's awesome. So it's a little bit after the hour, so I'm going to go ahead and close off here and thank both Gabe and Apoorva for their fantastic talks.